Okay, guys. Okay, so quick business. Exam 1 is next week. I am stopping, like, whatever we cover up till the end of lecture today is what's on the exam. I'm not going to test the people on Friday on something different than the people on Monday. You all have the same exam with the exception of the fact that some questions are different and the short answers will be different for sure because clearly I'm not dumb. So, so you have different exams and you could share as much as you want, but it won't help your friends. What else? Yes, so by now hopefully everyone started studying, and this is a gentle reminder that you should have started studying, because if you haven't, then you might be panicking. Come Monday, hello people, some people are taking this exam on Monday. So, 15 to 25 multiple choice. Oh man, I usually leave the door open. Sorry. 2 to 3 short answers. And there will be a quiz this weekend, but I will be nice to you, and it will be a material that will be on the exam, so you don't have to start reading something that's not even going to be on the exam. So I know I would be angry if a teacher did that to me. But we need the quiz because we need a certain number of quizzes so you can drop. So I'll make something up. Okay. So where were we? We talked about post-translational modifications and how they regulate protein-protein interactions or protein-DNA interactions, all to affect the cellular function. We talked about domains, and actually a student in Monday's recitation had a really nice, let's see if I can do this, had a really nice example or a nice way of thinking about domains, and I won't make her come up here. Let's see if I can figure this out. Up, go away, and which button should I press to make that go away? Oh, no show. There we go. Okay. Hopefully it comes back when we want it. So people from the questions I've been getting, I'm working it up. Okay. From the questions I've been getting, I'm thinking that most people still don't understand what a domain is. Hopefully the extra credit question helps people to understand it because you've had to blast something, find a different domain. Still, some people didn't give me that information in their answers, so that made me think they still didn't understand the domain thing. So we're going to try one more time, and I think a student who maybe I see, maybe I don't, who helped me with this the other day. So you guys can teach me how to teach you better. Okay, in terms of domains, because people come to me and they're like, okay, so a domain comes and binds this and simulates something, what's going on? And I'm like, no. Okay, so the way she said it is if you think about protein structure, and you've got, remember, you've got your N-terminus, and then eventually it folds into something, which might be an alpha helix, and then there's a loopy thing, and then maybe there'll be a beta sheet, and maybe there'll be some kind of linker into another alpha helix, and another alpha helix, and another alpha helix, and then maybe some beta sheet. Remember, what is this? A secondary structure and tertiary structures interacting together to form a protein, remember? Well, when I talk about domains, I talk about how maybe this chunk is one domain. This chunk is another domain. Maybe that chunk is another domain. This chunk might have one function. Maybe it's your ATPase domain. So it will bind to ATP. It's got some magical site here which binds to ATP and hydrolyzes it, right? ATP, ADP. With a bummer for those who didn't come today. They're not going to see this. Anyways, maybe this is your chromodomain. And so it will bind to, actually that's a bad place because it's up there. Make this your chromodomain. And it'll bind somehow to some other protein, right? Because chromodomains are points of interaction with some other protein which has its own structure. And it will bind because there's this acetylated lysine on it. And then this will be some other domain, maybe it's a DNA binding domain, and so it'll bind to the DNA. So you would say this protein has 3 domains, and each of those domains has a different function. Okay, does that clarify things for people? Yeah, uh-oh.
I would just like to say, like, it's not that each secondary structure is its own domain.
No.
It's like they work together. So you circle one out in the helix, but it can be working with other.
Yeah, I'm just doing it for simplicity.
Yeah.
Yes. So no, not each secondary structure is a domain. It's just that a functional unit, which may be formed by multiple structures that are close together, thank you for clarification, will form a domain. The domain has to do with its function rather than its structure, but of course structure relates to function, so there you go. Yes?
So if we say one of those is like a bromodomain, so that will bind an acetylated lysine, which will cause modification.
No, the acetylated lysine is the modification.
modification, which causes the change in.
Which causes, that's why it's able to interact without this bromo. So let's say this is a bromodomain. Let's say that's a bromodomain, right? And it's binding to the acetylated lysine. That's how those 2 proteins are interacting. If this was gone, they couldn't interact. And let's say these 2 proteins, let's say this is your histone, and this is your some protein that's like a nucleosome remodeling complex protein, or something that's going to bring in a nucleosome remodeling complex. So if this doesn't happen, this doesn't get brought in, then you don't change your chromatin structure. Okay, I'm trying to kind of connect everything for you guys. Yes?
So the domains are not something that binds to the protein, it's just different subunits of the protein.
It's different parts of the protein. So when I say it's a functional unit of a protein within a protein, it's actually like your functional unit would be this chunk of it.
But it's not attached to the
It
protein.
is. It's one long amino acid chain, right?
Right, but it's not something on top of that, it's just part of that protein.
It's one protein with many different chunks which do different things. Yes. Yes.
Do each domain have its own receptor on the histone and myosin?
There are thousands, probably, of conserved domains. Some have nothing to do with binding to histones. So every domain has a different function. Some, like the ones I talked to you about, are involved in protein-protein interactions. So a chromodomain only binds to methylated lysine on a histone. Make sense? Okay. Anything else? Okay. Well, I will move on. But I wanted to share that because I thought it was a really nice way to show it. Let's see if I can make this happen again. What happens if I do that? It's like a little bit of a dimmer. Okay, interesting. Ah, it came back. Okay, great. It could have been bad news. Okay, so domains. Concept of domains. Good, there, now everyone gets it. We talked about the enzymes that regulate chromatin structure and function. These are the enzymes that are making the modifications. We talked about the concept of the histone code and how multiple modifications regulate chromatin structure and function. And then you can think about the whole way of the way the book talks about it as there are these readers and writers, and the readers will read the histone code and recruit writers that will then modify other things. I think that's a really nice way to think about it. Give it a try. We talked about histone variants, and those are just different histone subunits that are added to histones under different situations. DNA methylation versus histone methylation, that's what we started to talk about. I want you to be sure to understand there's a difference between those two, a serious difference between the two. And then we were ending with the function of DNA methylation. Did I see a hand up in the air?
I was gonna ask what the difference with the sounds like.
I'll talk more about it, but we talked a little bit about, one is you're methylating DNA and one you're methylating histones, right? And there are different enzymes, different things that might happen, but similar because it has to do with recruitment of other molecules. But if you methylate your promoter, it's going to be very different than if you methylate a histone, right? Okay. So there were some questions that came up on Wednesday, some of which I can kind of answer, some of which I still can't answer, and I guess that's your lesson, that not everyone knows everything, and not everything is known. So, first question, this question was just, someone came up to me in the end and said, why cytosine modification and not some other base? And I was like, oh my god, I'm not sure, we need to think about this. I was emailed a nice article talking a little bit about cytosine modification, and how different modifications affect function, but it still didn't really talk about why cytosine and not some other base. And so I actually did a little bit of thinking and reading, and it really has to do with the structure of DNA is what I've come up with, and the availability of something to be modified. If anyone is a chemistry major and can think of this in a clearer way. So the way I understand it, here's your C, right? Here's your cytosine, and your modifications are happening here. Here's your GC base pair, here's your AT base pair, and your modifications usually happen here. And if you think about it, the rest of the stuff is all busy, right? And here's your AT, and there's not many places. I don't know about here, but there's not many places for modifications to happen because it's a nucleophilic attack, and we need to have electrons that are available for, I don't know. So I kind of came up with an answer, not so much. Yes.
Well, if you look at it, like, as if you're talking about down at the nitrogen end,
Mm-hmm.
the
There.
bonding.
There's nothing available,
Yeah,
right?
to break a double bond is very difficult. So if you look over, let's see where it binds on the C.
But over here, so here's where your modifications are happening on the
So
C.
it's easier to get rid of a hydrogen than it is to break it.
Right. Right. So that was my understanding, is that it's easier to mess with this over here than over here. And other than that, like a lot of it is busy binding to the other base. But I feel like I can't do much of a better job. What we need to do is get Dr. Weldon in here, because he also teaches biochem, and he could probably explain it much more clearly. But it's above and beyond what you need to know, but people ask me questions, and I feel the need to try to address them. Extra credit number 1. If DNA methyltransferase donates the methyl group from within its active site, is there a way to recharge the enzyme, or is it a single-use enzyme? I got a few answers to this, though none of them seem to answer the question directly. I got one which said that there's a methyltransferase there that then methylates the enzyme again. I couldn't find that information, but that was something that was sent to me. And everything else, I only got like 2 or 3 answers, which makes me think that if either people didn't look or they couldn't find it. And I spent a little time looking and I couldn't find it. That could mean it's not known. It could mean it is known and just not by me. So, yes.
I read something that said they don't really know much about it.
So you read something where it says it's not known. Yes.
I saw something about like some acidonicil or something that actually donates it, not the methyltransferase itself.
But I thought that was within the. So yeah.
I
Okay.
couldn't find anything.
Yeah. So yeah.
Well, I read that there was another adenosine transferase that also methylated adenosine, and there was a cytosine transferase as well. But I couldn't find anything about what happens after one or the other.
Okay. So it seems like it's inconclusive. Has anyone found anything that they feel good about? Yes.
Well, I didn't find anything, but I just, in my opinion, if it's single-use enzyme, we don't call it enzyme though, because the definition of enzyme is that it's catalyst that we can use over and over again. So if it's single-use enzyme, then it's not an enzyme.
So the comment is, if it's a single-use enzyme, then you can't call it an enzyme because the definition of an enzyme is something you can use again and again. Yeah.
It also seems pretty wasteful by the cell.
It's extremely wasteful. There's got to be a way. I don't think it's single-use. Okay. So we'll move on. So this remains a question because I really need to be convinced of something and understand it well to be able to teach it to you, and I've yet to find anything convincing. Okay, here's the next one. This was blast this and send me the, well, what's the likely function based on domain organization and what domains are present and what do they do. So you needed to tell me a likely function and the domains that are present and what they do. People sent me all sorts of things. Most of them did not include that information, so you can't get the extra credit because you didn't do the assignment. This is a lesson learned about reading or listening or clarifying what the assignment is and making sure you do it. A lot of people did send it and they will get the point. So what I found, so when you blast it, you get at the top of the page this really nice domain organization, conserved domain organization. And so what there is a HeLa-C, a DEX-DC. Those are both helicase, SANT, DNA binding, slide, DNA binding, and HAND, which is DNA nucleosome binding. So from that all together, you can make an assumption, right? That it's a nucleosome or chromatin remodeling enzyme, and if you then click on it, you'll actually be able to read about the protein, which is kind of cheating, but whatever, you want to find the information. So, any questions about this? A number of people sent me something about it being a sialidase, and I don't know where that came from. Yeah.
I blasted it and there was a Sanger website and that came up with a bacterial domain that was used for antiviral.
Oh, weird. So the Sanger website, huh? Yeah.
I think that was from lecture 5. The one that you put up on lecture 5, that same sequence, we confused it with the one that you put up for lecture 8.
Okay, so there was confusion. Okay. Well, let's move on. Okay, so we ended the other day on we were talking about the biological function of DNA methylation. Okay, so we already know the biological function of histone methylation, right? What's that? It's associated with heterochromatin formation. What else does histone methylation do, generally? It's involved with transcriptional silencing. But what does it do? What does the methylation of the histone tail do? It recruits a chromodomain containing protein, yes. And that is likely to do what? Alter structure and function of the chromatin, either in an activating or repressive way. It's often associated with repression, but sometimes activation. So now we're going to move on to DNA methylation. So this is not histone methylation, but methylation of the cytosine in the DNA. So in bacteria, methylation is used to identify the newly replicated DNA strand so that they can properly repair DNA after replication. Because methylation, it takes some time for methylation to happen, so you'll end up with one strand, your old strand, your parental strand will be methylated, and your new strand will not for a little while. So you can think the bacteria can identify which strand is the wrong one, right? If you have an error in your DNA replication, you don't want to repair the parental strand by mistake. So that's one thing that they do. The other thing is to identify phage DNA and destroy it. Right? Phage are viruses that infect bacteria, and it's kind of like the bacteria's immune response in a weird way. It's not methylated. Phage DNA is not methylated, only bacterial DNA is. So it could be recognized and destroyed.
Wouldn't eventually the phage DNA that was incorporated in the genome, wouldn't it become methylated eventually?
Wouldn't the phage DNA become methylated eventually? So maybe think of this as an innate immune response. It's not really an immune response, like the bacteria would have an immune response. I don't want to go there. But like when the phage come in, it takes time until you get integrated into the DNA. So this is the immediate recognition of phage DNA, immediate cleavage by the HHA1 endonuclease. Good question. Yes, I would guess that integration of phage DNA into the genome, that would be nicely hidden. Yeah. Okay, so that's bacteria. None of that happens, that does not happen in plants and mammals. In plants and mammals, DNA methylation is associated with silencing. So I want you guys to think about it. Just think about what silencing is, and I want you to propose a mechanism. So I'm going to give you guys a toolbox, and it's these few points. Think about the kinds of proteins that might bind to methylated cytosine. What kinds of proteins have we learned about? How might they have a silencing effect? Think about what's required for transcription to occur and how methylation might inhibit that. So stop for a minute and talk to your friends or neighbors. You don't have to be friends with your neighbors. You should be. It's good to be friends with your neighbors. We'll talk in a minute. Okay, are we done or do we want to have another minute? Does anyone have some kind of mechanism whereby DNA methylation might result in gene silencing? Yes?
Could the larger methyl group just provide a physical barrier for RNA polymerase?
Could the larger methyl group provide a physical barrier for RNA polymerase? Yeah? That's one? Yes? Methylated cytosine would recruit some proteins that would restrict transcription. Okay, so one is just a physical barrier. There's a bump in the road. So RNA polymerase can't bind. The other is a very large bump in the road is recruited. What else?
name the protein that binds that inhibits the promoter region?
A protein, right? So so a methylation within the promoter region could recruit either a protein or inhibit transcription itself. Yep. What else? Yes.
It could recruit a chromatin remodeling complex and close up the DNA.
It could recruit a chromatin remodeling complex and help to close up the DNA. Good. Yes.
I just got a question. Relative size of these polymerases compared to a methyl group, would that really be a significant bump?
Okay, so you're saying polymerase is huge,
Yes.
methyl groups are really small,
Extremely small.
extremely small in comparison. Would it be a real bump in the road? Maybe that's a bad analogy. Think about though how wait, do you have an answer to this
I have
or a theory?
statement.
Okay, so so let's see what the students have to say, and then I'll give you my thinking. Yes.
Could it just be a difference in charge? Say the polymerase couldn't function under certain conditions.
So will it make a difference in charge? So I don't know. I'm sure it will, but will that be the reason? I'm not sure. Yes.
Well, for one thing, you're not contending with the entire size of the RNA polymerase, you're dealing with the active site, so any kind of, I guess, if it runs in anything that's less than perfect as far as interacting with it, it would at least slow it down somewhat.
Okay, so it's not about the whole. It's not about the whole polymerase. It's about the active site. Well, I don't know because actually it's about. In my mind, it's about recruitment of RNA polymerase to the to the DNA. So the active site is kind of inside and not going to be affected so much. What else?
I'm just going to say that the polymerase is highly specific. It can recognize specific chemically similar bases, so it could have an effect.
Okay, polymerase is very specific. It recognizes specific bases. And mind you, I'm being extremely general when I say recruitment of polymerase. What I really mean is recruitment of all the transcription factors that then recruit polymerase. And that's highly specific. Does anyone have any other thoughts to share? Well, so it's transcription factors that bring in the polymerase, and those are extremely specific to what bases they bind. So in a way, if the charge or something is off in that region because of methylation of the cytosine, that's going to prevent interaction because methylated cytosine is often present in the major grooves of the DNA. And that is usually where your transcription factors are going to come and bind. So it messes with those molecular interactions. Yeah, bump in the road maybe is not the best analogy. Any thoughts about this? So those were all really good mechanisms. So clearly I don't need to teach you anything. Because you know what, you guys have the information, it's just about thinking about it and using it. And if this is difficult for you, reach out to your students, reach out to me, because sometimes you just need to figure out how to think about things. Okay, so here, yes?
For the histone methylation, it was sometimes activation and sometimes silencing, so is it the same person?
I would say with DNA methylation, it is. I never want to say always, but it is majorly associated with silencing. With histone tail methylation, it's different because you're not getting one modification at a time on histone tail. You're getting many, and it's that combination of modification that's resulting in recruitment of proteins that are doing things. Whereas DNA methylation, it's one kind of modification happening on each cytosine that's going to be methylated. It's a little bit different. I don't know if that satisfies you. So what can be done here? Okay, well, one thing that might happen is you might get methylation of DNA. Now, this is methylation of the DNA wrapped around the histone. It's not the histone tail. and it recruits a DNA methyl binding domain containing protein, which then will recruit an HDAC. And that HDAC is going to deacetylate the histone tails in the area. What does deacetylation of histone tails do? Silencing. So that's one thing that actually didn't come up in the proposed mechanisms. But right, recruitment of HDACs, right? Yes.
Can you say how it happens?
So you have your methylated DNA right there. See the blue triangles, methyl groups added to your cytosines on your DNA, and that results in recruitment of some protein that specifically recognizes the methylated DNA. So it has a DNA methyl binding domain, and that will recruit a histone deacetylase, and it will deacetylate nearby histone tail.
Now, how does it like influence? I mean, I guess proximity, but like how far does it reach?
How far?
Like, does the effect of the histone deacetylate reach? Like, does it depend on
It
the
has to be
size?
close enough to be near the active site. The question is, how far away can HDACs act from what distance? It has to be close enough to be in the active site. It's not going to go and deacetylate like 10 histones away. It's got to be close. It's got to be in the active site. Okay. Another thing that can happen is you can have recruitment of histone methyl transferases, right? So here you've got methylation of DNA, and then a histone methyl transferase will be recruited, and then that will methylate histone tails. And that is associated with formation of heterochromatin. Questions about this? Yes?
Is it like this will lead to the previous step?
These are all different things that can happen.
So they don't all happen at the same time?
No, I think that a lot of them will happen at the same time if you're trying to form heterochromatin, but it's not sequential. I'm not telling you it in any specific order. We're just going through all possible mechanisms of how of what DNA methylation might do. Yes.
Are these enzymes facilitated, or do it just happen by chance that these things find their spots?
Are these enzymes facilitated, or do they randomly find their spots? Well, for example, here you've got this protein that brings it in. I mean, everything, you're asking for a very specific level of regulation. Everything's highly regulated. You have to think that there's a certain amount of proteins in the cell. These are in the nucleus. They're chromatin associated or nearby, and then there's this constant movement, and it's going to eventually bind. Are there other proteins that help bring them in? I'm not 100% sure. These things do tend to happen in large complexes of proteins, but I can't tell you for sure. Yes?
So does that mean you can't have histone methylation without DNA methylation?
Does that mean you can't what?
Have histone methylation without DNA methylation?
No. So does that mean you can't have histone methylation without DNA methylation? No. It's just, I think the way to think about this is just that the different modifications can do many different things, and just think about all the things that tend towards silencing and all the things that tend towards activation. Think of it that way. You don't necessarily need methylated DNA to have a methylated histone. In fact, you might get a methylated histone that then results in methylated DNA. I know, I'm purposely vague. Sorry about that. So what else? Okay, so you can get recruitment of DNA methyltransferases. So you could have a chromodomain containing protein. Wait a minute. Wait a minute. That's the DNA methylation. Oh, okay. So you can actually have A chromodomain containing protein recruited to a histone tail, which will then bring in a DNA methyltransferase to then methylate DNA. So it goes both ways, just like the question we just got. And then you can also get recruitment of nucleosome remodeling enzymes. You can get methylation on your DNA, which then will be recognized by some sort of methylcytosine recognizing protein and recruit a nucleosome remodeling enzyme, which will help to form more densely packed chromatin. Any questions? Pretty simple, right?
You mentioned that you can have a binding which is strictly protein binding, and there is no like active site that it binds to. Like there's no like lysine that it's involved with. Is there one like that for this situation? Strictly proteins binding and not like with a specific site.
So there was a question: is there possible to be recruitment of some of a DNA methyltransferase by a protein binding to the DNA? Why not? I don't have a perfect example right now, but why not? So all this, so that's the thing, is like, think of the nucleus as, or the cell, as a toolbox full of different tools that can be used to do different things to get to a function. And so these modifications are all about recruiting different types of proteins to do different things. I know, it's really, I should be more scientific in the way that I say that. Okay, so here's an example of how a methylation of DNA is involved in establishing repressive chromatin. So this is tightly packed. Oh, did you have a question?
I was just kind of confused about like the big difference between DNA synthesis and methylation, because it kind of seems like we're recruiting the same things, and the end result is the silencing or closing of methylation. So what's
Yeah.
the
The big difference is one time you have DNA being methylated, and another time you have a histone being methylated. You're right, a lot of the same proteins are recruited, and the overall effects can be similar, but people confuse them a lot, and on exams tend to get them wrong. So I want you to really understand there's methylation of DNA, and there's methylation of histone tails. And so like you're not going to recruit a bromodomain containing protein or chromodomain containing protein to methylate DNA. Yes? With DNA methylation, how would you ever have gene expression? So the question is, how could you have gene expression if you've got all this methylation? Is that what you're saying? Demethylate. So think of it as a level of regulation. If things need to be silenced, sometimes things will be permanently silenced. And then you won't have you won't have gene expression. Let's say, think about the genome as this huge thing. It's this huge thing with many different genes. Some genes need to be expressed all the time. Some need to be expressed only during development. Some need to be expressed only in neurons. Some need to be expressed only during an immune response in T and B cells. So this is a way where you'll have permanent repression of genes that are never going to be needed in that cell type or in that phase of development. But it's also a way to silence things and then have them not be silenced. So, and that's why I say it's reversible, because you have methylation and demethylation. Any more questions, comments? Yes?
So I was wondering, so that means that the DNA methylation will completely silence the gene expression, not at different levels.
DNA methylation is highly associated with formation of repressive chromatin. And in that case, you'll have silencing of gene expression.
So it's going to be completely silenced.
Completely silenced. It depends on what level of methylation you have and what level of, whether you have heterochromatin, like very tightly packed heterochromatin formation, or not. But yeah,
So that means we could have gene expression with the presence of methylation.
when methylation is there on the DNA, you're not going to have gene expression. You have to remove those methyl groups. Okay. Now, histone methylation is different. DNA methylation, yes. Okay. Okay. So this is an example of how you can have these histone reader and writer proteins working together to establish repressive chromatin. So here's your chromatin, and you'll get some gene regulatory protein complex that's going to come and bind, and it's going to recruit some histone modifying enzyme. It's going to modify your histone tail, which will then recruit some other protein that's going to read that modification, and then modify something else, and that's going to spread down the chromatin. And then you'll get recruitment of a DNA methylase that will then come and start methylating the DNA. And so then you have this compact, you'll get this compact DNA, the recruitment of chromatin remodeling complexes that are bringing your nucleosomes closer together. And then you can end up recruiting methyl binding proteins that are going to do something else like further acetylate, further methylate, and that's how you end up with this repressive chromatin. Yes?
The reader protein and the DNA methylase enzyme, they can work at different speeds. They don't have to work at the same time.
The reader,
The green protein that's reading and adding on new.
the reader protein that's recognizing.
Yeah, the speed in which it methylates the histone can be different and is different from the enzyme that's methylating the DNA. Because
Yeah,
it's
you
just a
just
different
need to
rate.
get, it depends on when things get recruited, when they work. Yeah. Okay. Any questions? Okay. So let's bring this all together. I know this is seeming like a lot of stuff. Hopefully the fact that I've spent nearly 2 weeks on it makes it pretty clear. But I want you guys to be able to connect histone modification with recruitment of proteins, with activating or repressing activities, and DNA methylation, and how it has an effect on gene expression in response to some kind of outside signal. So we've got about 5 minutes left, so I'd like you guys to work in groups and draw out different scenarios of how these modifications are going to work to do different things. Okay? I know that might be really easy for some people, it might be really hard for people, but hopefully two people who have different abilities will sit near each other. Okay? Do you understand what I'm asking? Draw it out. Draw out your chromatin, draw out your modifications, recruit whatever protein. How is this going to have an effect on gene expression?
That way, it's just it's not one thing. It's not all of them. Then you're going to so you need to work together in order to get all that. When you read the things, you get like, you get a fundamental growth where you're like thinking about so while I was working on this last quiz, we looked at a whole bunch of things, so I wouldn't be able to tell you very really fast. Something like DNA methylation is more long-term silencing than histone methylation. I don't know. While we got to this question is about teenage Some people want to promote it, never. I realize I'm not in front of a great negative topic. Yeah. Well, different sample questions I wasn't but yeah, another one. I should see maybe some. I know what mistakes. Well, I can think of a trivial example, like the day after, you're like, well, give me a break with this. Changing textbook. But I should think I'm just trying to explain. Yeah. I found an example the exact opposite. Instead of interviews are not they get up and give I've got some PDF copies, and I look where everything seems to set centrifuge. And I saw like this is therefore the centrifuge not I can't remember the wording here. It's not directly influenced by it. Okay. We're talking about how arteries, regions, and exist other places in the genome, and that but also, you are passing the fact that that's where the DNA is already condensed there, and there are already proteins associated with it. So that's part of why the centrifuge is there, because the proteins are still that could cause spontaneous alternative to something. And then it's also the proteins that are associated with it that are causing it to be inherited to the like regions. So there are other regions that could have been somewhere where it could develop. So you could use like
That's why I'm saying bear with me.
One of the big things that I read, I got one, and then I look it up in the book, and I say, oh, all right.
Okay, guys, thank you. So, everybody, contact me with any questions.
I really appreciate it. $200 a day.